Hi, I'm Dr. Umar Malik, and in this video, we'll talk about acute inflammation. So, let's first start by understanding the basic concepts of acute inflammation. Suppose you get a cut on your skin. What do you think happens at the molecular level in that cut? Of course, some cells will be damaged there, and microbes like bacteria will be able to get in. In a reaction to this, the tissue and the blood vessels in that area develop a response that allows your body to contain the problem and get rid of the bacteria. How exactly your body does this will be discussed later in the video. So what exactly is inflammation? In simple terms, it's the body's response to harmful agents such as pathogens or damaged cells. The aim of this response is to bring white blood cells and defense molecules from the bloodstream to the affected area to eliminate the threat and begin the healing process. Before we proceed further, it's very important for you to understand the pattern of questions that have been asked from this topic in the last 10 years and how that pattern of questions has changed over time. During the 2012 to 2015 exams, the questions from this topic focused more on the basic concepts, distinguishing between acute and chronic inflammation and cardinal signs of inflammation. Then during 2016 to 2019, the type of questions shifted towards a deeper understanding of cellular mechanisms, mediators of inflammation, and morphological patterns of inflammation. But during the last 3 to 4 years, that is from 2020 to 2024, the questions have become more integrative, requiring knowledge of clinical applications, molecular mechanisms, and recent research topics. Hence, it's very important for you to focus on the latest trend of questions. So let's continue with our topic and try to understand the basics of inflammation. Inflammation is basically of two types, acute and chronic. Here are some important differences between the two. Acute inflammation develops rapidly, within minutes to hours, whereas chronic inflammation develops slowly, within a few days. The cellular infiltrate in acute inflammation is mostly neutrophils, whereas in chronic inflammation, monocytes, macrophages, and lymphocytes are the predominant cell type. Acute inflammation causes less tissue damage, which is usually self-limiting. But in chronic inflammation, the tissue injury can be severe and can even progress with time. Acute inflammation involves some prominent signs like pain, redness, raised temperature, etc. But chronic inflammation has less prominent signs. Acute inflammation basically consists of two main types of responses. The first involves the response of the blood vessels, and the second response is of the cells. Let's break down and try to understand what actually happens in both these cases. First, we will talk about the changes that occur in the blood vessels. Let's continue with our previous example of a cut on the skin. The blood vessels first undergo vasoconstriction, but then they quickly relax and undergo vasodilation. The initial vasoconstriction in acute inflammation is a brief reflex response aimed at minimizing blood loss and facilitating clot formation at the site of injury. It prepares the blood vessels for subsequent vasodilation, which increases blood flow and brings immune cells to the area. This phase is mediated by neurogenic reflexes and substances like endothelins and catecholamines. Like we just discussed, after this, the blood vessels quickly switch to vasodilation, which is the main response of the blood vessels in acute inflammation. This widening is what causes that redness and warmth you often see in an inflamed area, because now, due to vasodilation, more blood is being delivered to that area. In acute inflammation, increased vascular permeability is essential, so that immune cells and defense molecules can exit the blood vessels and reach the site of injury or infection. Several mechanisms contribute to this increased vascular permeability. The most common cause of vascular leakage is the contraction of endothelial cells, creating gaps that allow fluid to escape. This is triggered by mediators like histamine and bradykinin and is known as the immediate transient response, typically lasting for 15 to 30 minutes. In some cases, such as with sunburn or mild injuries, a delayed response can also occur, where vascular leakage begins hours later and lasts longer. The second mechanism which contributes to increased vascular permeability is endothelial injury. Now, direct damage to endothelial cells, which are the cells lining the inner wall of the blood vessels, can occur due to severe physical injury or microbial toxins. This leads to cell necrosis and detachment, resulting in sustained leakage. Neutrophils adhering to the endothelium during inflammation can exacerbate this damage, prolonging the leakage until the vessels are either thrombosed or repaired. Often, multiple mechanisms contribute to increased permeability in blood vessels, such as in thermal burns, where both endothelial contraction and endothelial injury play a role. This can result in significant fluid loss, which can be life-threatening in severe burns. The lymphatic system also responds to inflammation by increasing lymph flow to drain the excess fluid, leukocytes, and debris from the inflamed area. This can lead to secondary inflammation of lymphatic vessels known as lymphangitis or lymph nodes known as lymphadenitis, often marked by enlarged, painful lymph nodes. Red streaks near a skin wound can indicate lymphangitis, often signaling a bacterial infection. Now that we have got the blood vessels sorted, let's talk about what the white blood cells are up to during this time. The first responders here are neutrophils. Think of them as the body's emergency team. They start by moving from the middle of the blood vessels to the edges or the margins of the blood vessels, hence known as margination. Then they start rolling along the vessel walls, kind of like they are checking out where they can find an exit. This rolling action is controlled by special molecules called selectins, which are proteins attached to the neutrophils and endothelium of the blood vessels. Eventually, these cells stick firmly to the vessel wall thanks to special proteins known as integrins. Once they are stuck, the neutrophils can then squeeze through the gaps in the vessel wall, and this is called transmigration or diapedesis. And then they head straight to the site of inflammation to take care of the problem. Now let's talk about leukocyte adhesion molecules in a bit more detail. These are special proteins that are present on endothelium and leukocytes, and they lead to adhesion and transmigration of leukocytes. Here you have to remember about three main types of adhesion molecules. First, we have selectins. Two main types of selectins are involved, known as P-selectin and E-selectin. Selectins play a critical role in the initial weak adhesion of leukocytes, enabling them to slow down and roll along the vessel wall. On the white blood cells, we have attachment proteins known as integrins. Integrins lead to a more stable adhesion of leukocytes to the endothelium. Integrins attach to intracellular adhesion molecule or ICAM1, a key protein on endothelial cells facilitating firm adhesion. Different types of integrins are present on different types of WBCs, like LFA1 or lymphocyte function associated antigen 1, which is present on neutrophils. Then we have MAC1 on monocytes and VLF4 on T cells and monocytes. We also have PECAM1, also known as CD31. It plays a significant role in the process of transmigration, where leukocytes move between endothelial cells to enter the tissues. Remember, cytokines like TNF-alpha and IL-1 increase leukocyte adhesion molecules. All this has been discussed in much more detail in our beautifully illustrated, handcrafted, med-simplified notes. They cover every detail from this topic that you need to master and remember this topic for exams. Plus, our MCQ modules will help you to test your knowledge and prepare for exams confidently. So if you are interested, you can check the notes in the links provided in the description box down below. Genetic deficiencies of leukocyte adhesion molecules lead to increased susceptibility to bacterial infections. There are two important genetic defects associated with leukocyte adhesion molecules, which lead to a deficient immune system. These are leukocyte adhesion deficiency type 1 and leukocyte adhesion deficiency type 2. Defects in integrins lead to leukocyte adhesion deficiency type 1 or LAD1. LAD1 is characterized by recurrent bacterial infections, delayed wound healing, and absence of pus formation. One of the first signs of LAD type 1 is a delay in the detachment of umbilical cord stump after birth. Now, genetic defects in selectins are seen in leukocyte adhesion deficiency type 2. It also results in recurrent infections but with additional features like growth delay and Bombay blood group. The child also suffers from severe intellectual defects. A number of questions have been included from this section in latest exams. Let's review a couple of them. Question 1. A 2-year-old child presents with a history of recurrent bacterial infections and delayed separation of the umbilical cord. Laboratory tests reveal leukocytosis but no pus formation at the infection site. Which of the following is the most likely diagnosis? Here are the options. You can pause the screen and think about the answer for some time. The answer to this question is, of course, leukocyte adhesion deficiency type 1. Question number 2. A deficiency in which of the following leads to the inability of leukocytes to transmigrate across the endothelium? Here are the options. The answer to this question is PECAM1. So now we have understood about the basics of acute inflammation. Let's try to understand what happens after this. Continuing the example of a cut on the skin, suppose some bacteria get in through the site of injury. So it's important that your white blood cells spring into action fast. Imagine they are like a battalion of soldiers and they have to fight the enemy which has crossed into their territory. The enemy was spotted by the guard towers and the guard towers send a message over the wireless that they need help. With this example, let's try to understand what happens in your body when a similar situation arises. After exiting the bloodstream, leukocytes move toward the site of injury by a process known as chemotaxis. Chemoattractants are those substances that lead to chemotaxis. For example, the peptides and lipids present in bacterial cell wall act as chemoattractants. You can compare chemoattractants to the message that was sent by the guard towers to the battalion of soldiers, who, after receiving the message, know the exact location of the enemy. And the movement of these soldiers is the chemotaxis. For the purpose of exams, you have to remember some important chemoattractants which our own body produces. Cytokine interleukin 8 is a classic example. It guides neutrophils to the site of injury. Then we have complement system components like C5a, which is a powerful chemoattractant. Then we have arachidonic acid metabolites such as leukotriene B4 or LTB4, which is also a powerful chemoattractant. We'll study about the complement system and the arachidonic acid metabolites in our next video, where we will discuss the different mediators of inflammation. These agents bind to specific receptor on leukocytes, activating them to move towards the site of inflammation. The leukocytes move by reorganizing their internal structure, much like how a car with front-wheel drive is pulled forward by its wheels. Persistent neutrophilic infiltrate can be seen in infections like Pseudomonas, where neutrophils continue to be recruited for several days. In viral infections, we typically see an influx of lymphocytes rather than neutrophils. And in allergic reactions and helminthic infections, eosinophils are the dominant cells. Now we have a brief understanding of acute inflammation. Let's understand how our cells recognize harmful microbes and damage cells, which is essentially the first step in phagocytosis, which we'll discuss after this. Our cells have specialized receptors, like toll-like receptors, that act as early warning systems. These receptors are found on different parts of the cell, including the plasma membrane, endosomes, and cytosol, allowing them to detect invaders no matter where they are. Our cells also have node-like receptors or NLRs that detect internal danger signals like uric acid and ATP, which come from our own damaged cells. These receptors activate a protein in the cell known as inflammasome, which then induces the production of interleukin 1, a key molecule that recruits white blood cells to the site of damage. In addition to the primary receptors that directly recognize microbes, Many leukocytes possess other specialized receptors. One of the key types of these receptors are those that bind to the Fc tails of antibodies and complement proteins. Now, what does that mean? When a microbe invades the body, the immune system often tags it with antibodies or complement proteins. This tagging process, known as opsonization, makes it easier for immune cells to identify, bind to, and ultimately destroy these invaders. These receptors on leukocytes recognize these tags, promoting the ingestion and destruction of the microbes. Lastly, circulating proteins like those in the complement system and mannose binding lectin play a crucial role in recognizing and attacking microbes, further fueling the inflammatory response. Now that we have explored how leukocytes are recruited to the site of infection or injury, let's dive into the next crucial step: how these cells recognize, engulf, and then destroy these harmful agents through the process of phagocytosis. Phagocytosis is the method by which neutrophils and macrophages, the body's primary phagocytes, engulf and destroy microbes and dead cells. Here's how it works. The first step is recognizing the enemy. Phagocytes have specialized receptors that help them identify and attach to harmful agents and cells. We have discussed some important receptors which help WBCs to detect pathogens earlier in this video. In addition to them, mannose receptors present on macrophages is also one such example. With this, the macrophages bind to sugars like mannose and fucose, which are found on the microbial surfaces but not on human cells, allowing the phagocyte to distinguish between self and foreign cells. We also have something known as opsonins like IgG antibodies and C3b component of the complement system. We'll discuss more about these in a bit. But phagocytes have high affinity receptors for these opsonins, facilitating the process of recognition of pathogens which are coated with these opsonins. The second step in phagocytosis is engulfment. Once the phagocyte has attached to its target, it engulfs it by extending its cytoplasm around the particle, forming a phagosome, which is basically a membrane-bound vesicle inside the cell. The phagosome then fuses with a lysosome, creating a phagolysosome, where the actual digestion of the particle takes place. And the third step is intracellular killing and degradation. Inside the phagolysosome, the phagocyte unleashes a barrage of toxic substances to kill and break down the engulfed pathogen. You can imagine them as the weapons and bullets of the soldiers. In neutrophils, these enzymes and chemicals are stored inside special structures known as granules, which are of two types: specific or secondary granules and azurophilic or primary granules. Each type of granule contains a distinct set of enzymes and proteins that serve different functions in the immune system. Let's first talk about specific or secondary granules. These smaller granules are packed with enzymes that help in breaking down various extracellular components. They include lysozyme, which breaks down bacterial cell walls; collagenases degrade collagen; and gelatinase breaks down gelatin and other matrix proteins. We also have lactoferrin, which binds to iron and deprives bacteria of this essential nutrient. Then we have histaminase and alkaline phosphatase, which are important enzymes present in these secondary or specific granules. Next, we have the azurophilic or the primary granules. These larger granules contain more potent antimicrobial substances like myeloperoxidase, which is a key enzyme and plays a critical role in generating hypochlorite, which is a potent bactericidal agent. Means it kills bacteria. Azurophilic granules also contain a variety of bactericidal proteins like defensins, which disrupt bacterial cell membranes, and acid hydrolases, which break down various biomolecules within the phagolysosome. Additionally, neutral proteases such as elastase, cathepsin G, and non-specific collagenases degrade extracellular matrix components, contributing to tissue remodeling and sometimes tissue damage. Out of these, it's very important for you to understand the functioning of acid and neutral proteases. Acid proteases are enzymes that operate within the acidic environment of the phagolysosome, where they degrade bacteria and cellular debris. They are crucial for the digestion of engulfed pathogens. Neutral proteases, on the other hand, act outside the phagolysosome and can degrade various extracellular matrix components such as collagen, elastin, and cartilage. While this activity is important for tissue remodeling, it can also lead to tissue destruction during chronic inflammation. Neutral proteases also play a role in activating other components of the immune system, such as complement proteins and kinins, by breaking down their precursor molecules. The release of lysosomal enzymes into the extracellular space can cause significant tissue damage, especially if not properly regulated. This can lead to the amplification of inflammation and contribute to chronic inflammatory conditions. The body controls the activity of these powerful enzymes through antiproteases found in serum and tissue fluids. The most important of these is alpha-1 antitrypsin, which inhibits neutrophil elastase. Deficiency in alpha-1 antitrypsin can lead to unchecked elastase activity, resulting in conditions like emphysema due to destruction of elastic fibers in the lungs. Deficiencies in lysosomal enzymes can lead to buildup of undigested pathogens or substrates within the cells, contributing to a variety of lysosomal storage disorders. These will be discussed in much more detail in our biochemistry video series. I'll provide the links to all the related videos in the description below. Now let's talk about some topics that have been recently added in the textbooks of pathology. Cellular interactions and behaviors within tissues are complex and varied. Two phenomena that illustrate these interactions are emperipolysis and entosis. While they may seem familiar at first glance, both involve one cell being engulfed by another. Their mechanisms, outcomes, and clinical implications are completely different. Emperipolysis is a process where one cell is engulfed by another without being destroyed. Unlike phagocytosis, where the engulfed cell is digested and typically dies, in emperipolysis the engulfed cell remains intact and viable within the engulfing cell. This non-destructive interaction means that the engulfed cell can continue to function normally while residing within the other cell. The precise mechanism of emperipolysis is not fully understood, but it is thought to involve interactions between surface molecules of the engulfing and engulfed cell, allowing for a non-destructive engulfment process. Emperipolysis is an area of active research, particularly in understanding its role in disease processes and its potential implications in cell therapy and transplantation. In autoimmune hepatitis, emperipolysis is often observed in the liver, where lymphocytes, which are the engulfed cells, are found within the hepatocytes, which are the engulfing cells. This phenomena is considered a hallmark of the disease and can play a role in the immune response observed in this condition. Rosai-Dorfman disease is a rare disorder characterized by massive lymphadenopathy. In this condition, emperipolysis occurs when histiocytes, which is a type of immune cell, engulfs lymphocytes. This phenomena is one of the key diagnostic features of this disease. In myelodysplastic syndrome, which is a group of disorder caused by poorly formed or dysfunctional blood cells, emperipolysis can be seen in the bone marrow. The engulfment of hematopoietic stem cells by megakaryocytes is noted, although the exact significance of this in MDS is still under investigation. Entosis is a unique cell-within-a-cell phenomena where one cell actively invades another, leading to the engulfment of the weaker cell by a dominant one. Unlike emperipolysis, entosis results in the death of the engulfed cell, which is ultimately digested by the engulfing cell. Entosis is closely associated with the adhesion molecule E-cadherin. In normal tissues, E-cadherin helps to maintain cell-to-cell adhesion. However, in tumors where E-cadherin expression is often lost or reduced, cells lose their cohesion, allowing one cell to invade and engulf another. Once inside, the engulfed cell is encased in a vacuole and eventually degraded by the engulfing cell's lysosomes. This process can be viewed as a form of cannibalism, where the dominant cell consumes the weaker one, often to gain energy or nutrients. Entosis is most commonly observed in cancerous tissues, where it may contribute to tumor progression and survival. In aggressive tumors, cells exhibiting entosis may gain a survival advantage by consuming neighboring cells to meet their energy requirements. This phenomena can also contribute to tumor heterogeneity, making the cancer more resistant to treatments. Given its association with aggressive tumors, entosis is being studied as a potential marker for poor prognosis in certain cancers. It also raises interesting questions about the role of cell competition and survival within tumors. Entosis is a relatively newly recognized phenomenon and is a subject of ongoing research. Scientists are investigating its potential role in cancer metastasis, resistance to chemotherapy, and as a target for novel cancer therapies. While emperipolysis is generally non-destructive and seen in benign conditions or immune responses, entosis is destructive and is associated with malignancy. This stark difference highlights the diverse ways in which cells can interact with one another. In addition to their well-known roles in phagocytosis and microbial killing, neutrophils have another powerful weapon against infections: neutrophil extracellular traps. Nets are web-like structures composed of DNA and other nuclear materials that trap and neutralize pathogens at the site of infection. Let's break down how neutrophil extracellular traps are formed and why they are significant. Neutrophil extracellular traps are produced when neutrophils encounter infectious agents like bacteria and fungi, or when they are stimulated by inflammatory signals such as chemokines, cytokines, complement proteins, and reactive oxygen species. The process begins with the activation of an enzyme called arginine deaminase, which triggers chromatin decondensation. Other enzymes like myeloperoxidase and elastase then enter the nucleus and further loosen the chromatin structure. This process eventually leads to the rupture of the nuclear envelope, causing the chromatin to spill out into the extracellular space, forming a sticky meshwork that traps microbes. Reactive oxygen species plays a crucial role in activating arginine deaminase, leading to the conversion of arginine to citrulline and initiating chromatin decondensation. Then we have myeloperoxidase and elastase, and these enzymes further degrade the nuclear structure. The formation of neutrophil extracellular traps result in the death of the neutrophil, as its nuclear contents are expelled to form the trap. Let's look at the function and importance of neutrophil extracellular traps. Once formed, these traps act as a physical barrier, concentrating antimicrobial substances like peptides and enzymes at the site of infection. This helps to trap and kill the invading microbes, preventing them from spreading to other areas of the body. However, the process isn't without its drawbacks. Nets have been detected in the blood during conditions like sepsis, where they can contribute to systemic inflammation. Additionally, the release of nuclear material into the extracellular space can potentially trigger autoimmune responses. In diseases like systemic lupus erythematosus, the DNA and proteins in neutrophil extracellular traps might act as autoantigens, leading to an immune reaction against the body's own tissues. Acute inflammation presents with characteristic morphological features, most notably the dilation of small blood vessels and the accumulation of leukocytes and fluid in the tissues. However, depending upon the cause, severity, and location of the inflammation, several distinct morphological patterns may emerge. Recognizing these patterns is crucial because they often provide valuable clues about the underlying cause of the inflammation. Serious inflammation is characterized by the exudation of a cell-poor fluid into the spaces created by cell injury or into the body cavities such as peritoneum, pleura, or pericardium. The fluid typically lacks microbes and large number of leukocytes, making it less dense and less likely to cause pus formation. This type of inflammation is often seen in conditions like burns or viral infections, where fluid accumulates under the skin, as in blisters, or in body cavities, a condition known as effusion. When there is a significant increase in vascular permeability, larger molecules such as fibrinogen escape from the blood, leading to the formation of fibrin deposits in the extracellular space. Fibrinous inflammation is commonly seen in the lining of the body cavities, such as the meninges, pericardium, and pleura. If not cleared, fibrin can lead to scarring by stimulating the growth of fibroblasts and blood vessels, potentially resulting in the thickening of tissues and the obliteration of spaces, such as the pericardial space. Purulent inflammation is defined by the production of pus, which is an exudate consisting of neutrophils, necrotic cell debris, and fluid. This type of inflammation is typically caused by pyogenic, pus-forming bacteria like staphylococci, which lead to tissue necrosis. A common example is acute appendicitis. Abscess, which is a localized collection of pus within tissues, organs, or confined spaces, often form as a result of bacterial infection. These abscesses have a central area of necrosis surrounded by preserved neutrophils, with surrounding areas of vascular dilation and fibroblast activity indicating chronic inflammation and tissue repair. An ulcer is a local defect or excavation of the surface of an organ or tissue produced by the shedding of necrotic tissues. Ulceration typically occurs in areas where tissue necrosis and inflammation exist close to a surface, such as the mucosa of the mouth, stomach, intestines, or the skin of the lower extremities. Peptic ulcers of the stomach or duodenum are classic examples, where both acute and chronic inflammation coexist. The acute phase is marked by intense neutrophil infiltration and vascular dilation, while the chronic phase involves fibroblast proliferation, scarring, and the accumulation of immune cells like lymphocytes and macrophages. So this was all about acute inflammation. I hope you liked the video. I provided links to Med Simplified notes in the description box down below, so make sure to check them out. And yes, make sure to hit the subscribe button below because we are going to upload a lot of videos in the coming time. Thank you so much for watching.